Hello, everyone, and welcome. Um, Brian, could you give me a thumbs up if my audio is coming through? Thank you so much. Welcome and Jai Jinendra to all of you who are joining us today for the first inaugural Shri Parshvanath Presidential Chair in Jain Studies lecture in Ethical Innovations. My name is Brian Donaldson, and I hold the chair in Jain Studies here at UCI in the program of Religious Studies and Department of Philosophy since last fall, 2019. As we begin, just a note that at the bottom of your screen, you'll see a Q&A box. And throughout our time, please feel free to use that to enter questions as they arise in your mind. We will be having a conversation at the end of Dr. Ingber's talk, and Dr. Brian Cummings from the School of Medicine at UCI will be moderating those questions, and we'll try to get to as many as we can. I also want to thank Joanne Jamora and Sam Carter from the Center for Medical Humanities, who will help me to answer any basic procedural questions that come in through the chat. Thanks to you both. And finally, this lecture is being recorded, and registered attendees will receive a link in the next week on how you can access that. I'd like to offer now a few words of introduction. Jainism is a religious tradition and a philosophical system rooted in India, whose texts and practices are perhaps more robustly focused on the ethics of life and death than any other tradition in the world. In fact, the music that you heard playing as you joined today is called the Namakar Mantra. It's a Jain homage to 5 groups of inspiring persons who restrained their mind, speech, and bodily actions so as not to harm other living beings. Jainism posits a core life force in every living being, whether that's a human or an insect, an animal, or even a microbe. Consequently, the Jain tradition advocates nonviolence, or a word you might have heard, ahimsa, in order to minimize harm to all life. This sentiment is expressed in the oldest surviving Jain text, dated back to the 5th or 6th century BCE, which states the following: all breathing, existing, living, sentient beings should not be slain, nor treated with violence, nor abused, nor tormented, nor driven away. This is the pure, unchangeable, eternal law. This powerful exhortation not to harm is expressed in the lives of Jain monks and nuns who seek to avoid violence to all beings in as many ways as humanly possible. They give up family and professional life in order to avoid the accumulation of material goods and wealth that's associated with that. They don't wear shoes or use mechanized transportation. They wear mouth shields to prevent inhaling microorganisms. They eat simple vegetarian food prepared by others. They even use a broom to sweep the ground clear of insects when walking, sitting, or lying down, among many other iconic practices. However, the vast majority of Jains in the world are not monks and nuns, but they strive out to live out this commitment to nonviolence in the context of their work, their families, and their communities. Throughout the tradition's history, lay Jains have gravitated toward occupations that aim to inflict less harm, which came to include medicine. Today, Jains have high representation in medical and allied fields, including Jains who live here in the US. Today's lecture in ethical innovations emerges from my own research in Jainism and bioethics, including how Jain medical professionals strive to maximize nonviolence while grappling with how to ethically manage healthcare harms, such as injury in animals through testing, research, and dissection. Also, in teaching bioethics at the university level, many of my students have also expressed their moral struggle with this aspect of their education. Rather than silence this difficulty, I believe that we can do better at UCI, as one of the preeminent research institutions in the world, to bring this issue to the light of ethical reflection in hopes that we can join with others seeking innovative both and solutions that promote a thriving community of both humans and other living beings whenever possible. It's my hope that this lecture series will promote areas of common cause and collaboration between the Jain community and other campus supporters, and also UCI research communities in bioengineering, biosciences, medicine, and the humanities, where students, teams, faculty, and projects can be supported toward innovations that boldly strive for the well-being of as many lives as possible, both human and far beyond. It's now my pleasure to invite Dr. Jaswant Modi to offer a few remarks as a member of the regional Jain community. Dr. Jaswant Modi is a specialist in internal medicine and gastroenterology who has practiced medicine for nearly 5 decades. After completing medical school in India, Dr. Modi came to the US in 1975, where he first studied in Chicago, and since then he spent most of his career in and around Los Angeles. Dr. Modi is a founding member of the Jain Center of Southern California, and he and his wife, Meera, who is also a physician, are active benefactors in many international medical programs, such as Shankara Eye Foundation and Jaipur Foot, as well as supporting endowments at US universities for the development of the academic study of Jainism. I've invited Dr. Modi to say a few words on what has motivated his and the Jain community's support of this particular lecture series on alternatives to animal testing. Welcome, Dr. Modi, and you can unmute your mic and offer your remarks.
Can everybody hear me well? Is everybody able to hear me okay?
We can hear you, yes.
Okay. Jai Jinendra, and thank you very much, Dr. Briani, for offering remarks on my background. Welcome to all the participants, Dr. Ingber, and everybody else in the audience who are here to this special lecture. I, being a physician and practicing internal medicine and gastroenterology, I've been exposed to various forms of the hinsa or a violence that you can call right from my days with the medical school. I was born into a Jain family, and we had very strict habits, dietary ritual, and so on and so forth. So when I went to the medical school, right in the first year of medical school in anatomy, we were asked to dissect frogs, worms, and so on and so forth. And believe me, it was very difficult for us to do it. I particularly was thinking to maybe quit medical school and go somewhere else and do something else, which I didn't do it. But I'm here now, and in a medical field and many other fields, we have been exposed to different kind of a hurt, which would be a violence, subtle or obvious. And through this lecture series and various other technological improvements, we want to see if we can make society a better, where there's a less forms of the violence is instituted or committed to various forms of the life, including, of course, human beings, but animals, plants, and etc. And in that endeavor, we joined the hands with the UCI and Dr. Donaldson. And with this lecture, Dr. Ingber will enlighten us more to see how we can proceed and we can educate our society and various disciplines in a better way. So thank you very much. I appreciate.
Thank you so much, Dr. Modi. I really appreciate you being here on behalf of the Jain community, and to all of the local, regional, and national, international Jains here. Welcome, thanks for being here. Now we'll move on to our speaker. Dr. Don Ingber is the founding director of the Wyss Institute for Biologically Inspired Engineering at Harvard University, as well as the Judah Folkman Professor of Vascular Biology at Harvard Medical School and Boston's Children's Hospital. He's also Professor of Bioengineering at Harvard. At the Wyss Institute, Dr. Ingber's team works to develop bio-inspired technologies to advance both healthcare and sustainability, while blurring the boundaries between biology, art, and design. Their work has led to major advances in mechanobiology, tissue engineering, blood vessel formation in tumors, and nanobiotechnology to address blood toxins, among many other innovations. In addition to authoring in excess of 400 publications, Dr. Ingber has over 150 patents. Today, Don Ingber will be discussing his development of human organ chips created with microchip manufacturing methods and lined with living human cells as a way to replace animal testing as a more accurate and affordable platform for drug development and personalized medicine. The human organ chip technology was named Design of the Year in 2015 and included in the top 10 emerging technologies of 2016 by the World Economic Forum. And the next time you're in New York City, you can also find the human organ chip in the Museum of Modern Art, where it's a part of the permanent design collection. Welcome to UCI, Dr. Ingber.
Thank you. I'm going to share my screen.
You're coming through loud and clear.
Okay, I'm going to share my screen now. And can you see that?
Looks great.
Okay, great. So thank you so much for the opportunity to join you. And it's a high aspirations to meet the challenge of what the Jainists have valued for so long, but it's a great goal. I hope to show you sort of a baby step along the way, but the goal certainly is to progressively replace animal testing in medicine and other areas. And so I'd like to show you what we've done over the past 10, 15 years in this direction. So the title of the talk is Human Organ Chips from Experimental Models to Clinical Mimicry. And this is our Wyss Institute logo over the top. Those are all various innovations that have come out of the Institute. I've been a professor at Harvard for 36 years, and the last 11, I've been the founding director of the Wyss Institute. And the basic concept of the Institute is that engineering has transformed medicine and the environment over the past 50 years by taking engineering principles and trying to solve problems in medicine and sustainability. We basically tried to flip that on its head by realizing that we've uncovered so much about how nature builds, controls, and manufactures from the nanoscale up, that we feel that we're at a point where we could leverage biological principles to develop new engineering innovations. And that's what we call biologically inspired engineering. And we were lucky enough to get kickstarted with the single largest gift in Harvard's history at the time of $125 million from Hans Wyss, which we've tripled since then. But when he gave that money, he wanted us to take on really hard problems that were high risk but potentially huge payoff that NIH or NSF would not fund. And the biggest problem that I could see at the time was that the drug development model is broken. Industry currently spends probably over $3 billion a year to go from a discovery at the lab bench to an approved drug. There are many reasons for this. Some are that we do a lot of work with cells cultured in dishes that we know don't function anything like our bodies. The regulatory agencies require animal studies. They take years to complete. Innumerable animal lives are lost, and the ethics of that are extremely challenging. But they're even more challenging because the results often don't predict clinical responses, and some have estimated this to be over 90% of the time in certain areas. As a result, there's a lack of new drugs reaching patients, and there's been a search for better models that could actually mimic human organ level function, not cell or tissue, but organ. So when we started the Institute, we had 6 major platform areas, and I led one myself that has led to what we now call human organs on chips. The basic idea was to engineer microchips containing living human cells that reconstitute organ level functions, not cell or tissue, to accelerate drug development, to replace animal testing, and advanced personalized medicine. Now, why microchips? Well, microchip manufacturing offers control of surface features at the same nanometer to micrometer scale that living cells live at. And this is accomplished with photolithographic etching techniques. That is the basis of computer manufacturing. Now, over 25 years ago, George Whitesides, who's at the Wyss Institute as well, and I started to adapt lithographic techniques to create engineered cell microenvironments. George, well, if you go to how computer chips are made, you have a silicon wafer, a polymer resist layer, and then you could pattern it. On the computer, you design a pattern, and when you expose light in that pattern, only small regions, for example, the circles, are dissolved, and then you could etch the chip through that, and you can get a patterned chip. George developed an inexpensive way to do this where you can design a pattern, you put a photoresist polymer like that, but now when you make the holes, you pour a polymer on it that's liquid. It's silicon rubber called PDMS, and when it solidifies, you can peel it off, and you retain the surface topography down to 60 to 90 nanometer resolution. Then you could use that to stamp self-assembling monolayers or chemical inks, matrix molecules into patterns. Then you could surround with non-adhesive materials. And with that, we showed in a series of science papers in the early 1990s that if you coat it with matrix, you can control cell shape, function, directional motility, stem cell switching by controlling the local microenvironment. George started to make systems that are hollow channels with inlets and outlets that are called microfluidics. And this is what has been used to miniaturize all sorts of instrumentation, particularly for diagnostics industry. To me, this is really an engineered microvascular network. And so we put this all together to create what we call organs on chips. And the first breakthrough was about 10 years ago. We published in Science. We called a human breathing lung on a chip. Now you can think of these as living three-dimensional cross sections through a major functional unit of a human organ. So we're not building a whole organ. We're trying to build the minimal unit that retains organ level functions, but will also provide a window on molecular scale activities inside living cells in a tissue and organ context. So we started with modeling the alveolus, or the air sac. And for those of you who are not familiar, this is where, gas is exchanged, aerosol-based drug delivery, pneumonia, et cetera. And if you cut it in cross-section, it's a hollow structure like a bunch of grapes. If you go to the EM, electron microscopic level, there's air, there's an alveolar epithelial cell, there's a porous, flexible basement membrane matrix, extracellular matrix, and then there's the capillary endothelial cell, and there's blood. And what you don't see is that this is an incredibly mechanically active structure. Oops, I'm sorry. In that every time we breathe in, the cells, the matrix stretch, and when we breathe out, they retract. Plus, there's air flowing in and out, and there's blood flowing. Now, we know from respiratory physiology that these mechanical motions are absolutely critical for lung development and function. So if I was trying to distill down, what are the minimal design principles that makes this organ an organ? The first thing is that an organ are two or more tissues that come together and interface and new functions emerge. One of them often being a vascular tissue. And in the lung, we felt that these physical motions were critical, including dynamic flow and cyclic breathing motions. So this next slide is an animation of how this works. At the top right, you see a picture of the chip. It's the size of a computer memory stick. It's made out of that optically clear silicon rubber. Cut it in cross section. There are 3 hollow channels, each less than 1 millimeter wide. The middle channel is broken to top and bottom with a thin rubber membrane with engineered pores. We coat that with extracellular matrix. To make the alveolus chip, we have lung alveolar cells from human on top, lung capillary cells on the bottom. We just recreated the alveolar capillary interface. Now, the trick is we have cyclic suction in these side chambers. Everything's flexible so that now you get stretching of the cells and the membrane at the same rate and degree as when we all breathe in and out. You could put air over the top to have an air-liquid interface, and then you can have medium with or without immune cells flowing through the bottom. We now put endothelial cells on all 4 sides, and we could even have blood for extended time, for short times. So if this were to work, it should model organ level responses. So imagine you have a bacterial infection. In your lung, you get tissue signaling. Cytokines are released that activate the endothelium. Normally white blood cells flow by, but when they're activated, they put out things like ICAM on their surface. The cells stick, roll, diapedese across, and engulf the bacterium. Now you're going to see living cells in the device. These are fresh human white blood cells. We know they're fresh. We took them out of my postdoc, Dan, huh? You can't see the endothelium. It's not labeled, and the epithelium is behind the screen. To begin with, control, they just flow by like in a healthy blood vessel. We put bacteria on the other side. The signaling occurs, ICAMs expressed, these white blood cells are pulled out under flow, and that initial event is known to be shear stress dependent, the adhesion event. Now you can do any imaging you can do in vivo or in vitro. So this is high power. That's one neutrophil going between two endothelial cells that are unlabeled, finds the matrix, wiggles its rear end out of focus through that hole, coming to the other side. On the top, look by phase, you see the cell popping up between the epithelial cells. Now you're going to see the white blood cells in red because the bacteria are in GFP green, and you could see them being engulfed. So you just watch the entire human inflammatory response at high resolution in this little rubber chip. Now, we started to talk to pharmaceutical companies, and they said, this is great, but we're really interested in disease models and also toxicity models. So we hit two birds with one stone by taking the chip and perfusing the vascular channel with interleukin-2. It's an FDA-approved cancer drug, but its dose-limiting toxicity is pulmonary vascular leakage, leading to pulmonary edema. We gave it at the same dose through this intravenous route. At time 0, this is what the chip looks like with air. When you infuse it, you see a meniscus of fluid in the airway channel. Over 2 to 3 days, that completely fills that channel. And you also see blood clots there, which I'm not showing. And this is exactly what you see with the same dose and the same time course in patients with this toxicity. We could quantify this using fluorescent inulin. It's a trick from the kidney physiology world. And what was interesting is that if you have the breathing motions, physiological breathings, that's this top dark blue. If you have static chip that's more like a transwell, you do not see this toxicity response. Now, this is not mimicking human physiology. This is a prediction. No one's ever seen this before because you can't control this in vivo. So we did an ex vivo ventilation perfusion model in a mouse, and where you can separate breathing from the heart pumping, and we got the same exact response. Now, I've worked in this area of mechanobiology since I was 18 years old. And I think was one of the first to present the idea that mechanical forces are as important as chemicals and genes for developmental control and biological regulation. And I'd actually worked on what is the fastest signaling event that occurs when you stretch cells on a substrate, and in endothelial cells, that was forces going through matrix, through integrin receptors, to an ion channel in the focal adhesion called TRPV4, causing calcium influx in 5 milliseconds. So the finding that these breathing motions, the cyclic stretching, was so important made me seek out if I can get an inhibitor of that pathway. And I knew that the pharmaceutical company GlaxoSmithKline had a drug they were trying to develop. They were having problems. It wasn't really going anywhere. So I thought maybe I could get it, and I was able to get it. They didn't fund my work, but we were able to show that we could completely inhibit pulmonary edema in this model using that drug. This inspired them to go on and do a similar study in animals and dogs and rabbits using a model of cardiogenic pulmonary edema, where the heart failure gives you back pressure, so a different mechanism, and they completely inhibited pulmonary edema in these animals. And that is now in phase 2 clinical trials. And it's, I think, an example of how this has helped accelerate drug development already. So this one model, this lung alveolus chip, has provided proof of principle for a disease model, drug toxicity. And we also had another paper where we were able to mimic a toxicity that was not seen in animals. The drug company Janssen had a biologic therapeutic antibody for lupus. No toxicity in animals ever. Went to human clinical trials. Multiple people died due to pulmonary thrombosis. We can model that in the chip. We've also been able to model drug efficacy. We discovered new therapeutic target, and this has helped get drugs into phase 2 clinical trials. we've also integrated immune cells, as you've seen, and we haven't published in the lung, but we've integrated stromal fibroblasts to look at fibrosis as well. So this chip, as you heard in the intro, this has won numerous awards. I am most proud that we won the International Design Award a few years ago. We beat out the Google car and the Frank Gehry building. My wife is always most proud that it's in the permanent collection of the Museum of Modern Art. I'm a permanent member, which means I get a discount at the gift shop and the cafeteria for life. So she's very excited, as am I. And it was picked out as one of the emerging technologies by the World Economic Forum. Okay, so the drug companies came back and they said, well, this is really great, but most of the drugs we're working on are for asthma and chronic obstructive pulmonary disease, COPD, which are diseases of the small airway, not the alveolus. So we took the chip, we made the channel 1 millimeter high, which is sort of the radius of a bronchiole. We use primary human bronchial epithelial cells that normally are mucociliated. We put them on the chip under air for 3 weeks, feeding them only from the vascular channel, which is what it is in vivo. And at the end of that time, this is what they look like. They're ciliated epithelium. They actually move in a directional way. This is an EM picture, colorized, but beautiful. If you add fluorescent microbeads to measure mucociliary clearance, this is a real-time movie. And this is exactly the same rate as in our lungs. So it's like a little living small airway on a chip. Now, we then made chips with cells from patients with chronic obstructive pulmonary disease. What was amazing is that after 3 weeks of differentiation on chip, they retained the clinical phenotype, which is known to be associated with decreased TOLR receptors. And again, there are no inflammatory cells here, so this is just the epithelial cells. Now, what brings these patients to the emergency room are usually exacerbations by bacterial infection, viral infection, or cigarette smoke. We mimic this with poly-IC to mimic viral infection and LPS endotoxin. And what we could show is indeed that, for example, the COPD patient chips increased IL-8 secretion in response to a bacterial mimic, but the healthy did not. Whereas they also increased MCSF in terms of a viral infection mimic where the healthy did not. And this turns out never to have been seen before, and it could be a clinical marker to discriminate between bacterial and viral infection. Now, the other thing that brings patients to the emergency room is cigarette smoking. So we developed a cigarette smoking robot. That's a cigarette lighter from a car, a Gatling gun with real cigarettes. We could model the frequency, puff intervals, pauses, and real smoke goes right to the airspace of the chip. And when we did that, you can see, as shown at the left, and this is just a couple of examples, that normal chips with normal cells from a healthy patient, IL-8 does not change when you see cigarette smoke, but COPD double IL-8. And we now do multi-omics on these chips. So this is transcriptomics at the right. We've done proteomics, metabolomics, lipomics, et cetera. But here, what you see at the right, the 3 lanes at the right are 3 different chips from 3 different healthy donors. The change in gene expression relative to after versus before exposure to cigarette smoke. On the left are 9 patients' transcriptomic results from a past publication, healthy people who are smokers. You can see the general pattern is very similar. Top quarter versus bottom 3/4 is very, very similar. But the reviewers said that this shouldn't be published because our 3 are so similar and we didn't have the range of expression, et cetera. And then we realized that in some ways we could do studies better than clinical studies, and that we were doing true matched comparative modeling of the same patient before and after a stimulus. So these changes are related to smoking and not, where they worked and other habits and what they ate and so forth. And we actually got it in and were asked to do a little review on matched comparative modeling in vitro. Now, having developed these chips, we went back to look at cancer, which is what I started my career in and done a lot of work on. And in animal models, people knew that for years they modeled cancers by injecting subcutaneously, and they looked nothing like in patients. So then they moved to what are called orthotopic models, where in a mouse, they'll put a breast cancer in a mammary fat pad or a prostate cancer in the prostate gland, and they look a little bit more like in patients. So we have these engineered human organ microenvironments. So we developed human orthotopic lung cancer models. Excuse me, we started with a a non-small cell lung cancer line that's an adenocarcinoma form of this disease that in patients emerges in the distant bronchiole, but the cells almost always preferentially grow in the alveoli in vivo. So we plate the cells at low ratio to healthy at the time of plating. And here you can see in the alveolus chip, the cancer cells in green actually might grow in the epithelium and actually invade it into the blood vessel below. Now, when we quantitate growth, it was very interesting in that if you look at the small airway, which is where these emerge but don't grow in vivo, they grow very slowly in black squares, and then they start growing later on, again at a slow rate. If you put the cells in the same medium that we use on chips, but on a plastic dish, they don't grow at all. So all the growth is due to the microenvironment. If you put the same cells in a plastic dish with this rich serum containing medium that are used for passage, they grow extremely rapidly. That's what this line is. We put them in the alveolus chip in this medium that, again, they won't grow in open circles on a plastic dish. They grow much more rapidly than in the airway. And what was interesting is that we could show that breathing motions regulate tumor cell growth in that if you basically suppress growth by 50% with breathing, which could explode, if you think about it, and actually I should show you that it also suppresses invasion by 50%, migration through, which is easily measured by watching the GFP labeled cells pass through the pores. It also regulates therapeutic response to first line drugs like tyrosine kinase inhibitors. Here you see that without breathing, the tumor cells grow rapidly and you inhibit that growth. With breathing, the tumor cells grow slowly and there's no change with drug. And we could show this is regulated at the level of EGFR receptor signaling. But this can explain in part why, if you imagine you have a tumor growing and just beginning to grow in your lung, you're breathing in and out, it fills an alveolus that rigidifies. If this is right, that's a positive feedback loop. That will start growing more, fill the next alveolus and the next. Now, if you treat with drug, it'll shrink back. But once it begins to aerate again, now you have residual tumor again that is less responsive to drug. And this is kind of consistent with what we see in vivo. But more importantly, it provides a model to study residual tumor and dormant tumors. We then went on to other organs. So we made a model of the human intestine chip. We started with human CACO2 intestinal epithelial cells that are an established cell line. For 50 years, in growth medium, they grow as flat cells, either on a dish or in a transwall, even though in vivo they're columnar and form villi. We put them on our chip with peristaltic-like deformations as shown at the top and trickling-like flow like in your gut. In 5 days, they form intestinal villi, as you can see there. Now we work with cells, primary intestinal cells from organoids from patients. We take those organoids, we break them up, and then plate them in the chip. And that's what you see at the bottom from above or from the side. And the green are proliferative cells at the base, and you get these beautiful villus structures forming that with all the differentiated lineages of the intestine. Moreover, if you do transcriptomic analysis, what you see is if you compare the organoid that comes from the duodenum, and the right is the patient specimen, the left is the organoid, and the middle is our chip made from the organoid cells, we are much closer to the duodenum than an organoid. Plus, you can access both sides and you can measure transport, absorption, and also mucus production. So we made a colon chip. The top right is a cross-section through the chip that looks just like in vivo. We can put the chip on its side and watch mucus accumulation with DIC optics and live imaging, see it accumulate. And for the first time, we were able to show that we could reconstitute the classic mucus bilayer structure that's seen in human mucus of a similar thickness as in human colon mucus. Now, one of the biggest paradigm shifts in medicine in the last 20 years has been the microbiome. And we now know it's important for so much of health and disease, but almost everything we know is due to metagenomic studies, because if you put microbiome on a culture of human cells, we call that contamination and they die. However, with chips, we can mimic the in vivo microenvironment. And what we did is we literally created a hypoxia gradient across the endothelium and epithelium by having a chamber where we have nitrogen gas around the chip. We feed oxygenated medium as oxygenated blood through the vascular channel, and we get a gradient across. And we have oxygen sensors, and we can get below 0.5% O2, which means now we could grow obligate anaerobes and aerobes in the same time. And we actually confirmed this works. We can show growth of anaerobes and aerobes, and under these anaerobic conditions, we actually get better barrier function with the microbiome there. Now we've quantified this, and this is all published. And at the right is a movie that you might be able to just feel all of those little speckles or bacteria. That's the density of microbes on the chip. Those are villi. This came from a patient stool specimen, a preterm infant. And the level of complexity of what we grow on these chips for multiple days is over 200 different operational taxonomic units, or different strains of bacteria from 11 different genera that are similar complexity to stool. So we now have, for the first time, the ability to culture human and epithelial cells in contact with human complex microbiome for extended periods of time in vitro. Other examples, we made a kidney glomerulus chip. To do this, we had to develop a way to engineer induced pluripotent stem cells to form mature kidney podocytes. Kidney podocytes sit on one side of the basement membrane, endothelium on the other, and that's the filtration boundary. I'm not going to go into it, but we developed a way to do this. And now when you put the podocytes on one side, endothelial cells on the other, and you give it 1 beat per second of cyclic strain, 10% deformation, which is what people have visualized in living animals, the glomerulus does every time your heart pulses, then you could see the foot processes of the podocytes go up through the pores in the membrane and touch the basement membrane, and then you could reconstitute urinary clearance on a chip that's the same as in vivo. We also developed a blood-brain barrier chip by engineering human IPS-Derived human brain microvascular endothelium. We have human pericytes and astrocytes on the other side, very highly differentiated. But the cool thing is that for the first time, we get barrier integrity that is measured in ohms centimeters squared at the level of 4 to 8,000 or 4 to 7,000, whereas all those papers in the past were in the order of, our own included, were under 100. And this is similar to what you see in vivo. And we could also reconstitute shuttling of antibodies via transferrin receptors, which is one of the paths that people are exploring for shuttling therapeutic antibodies into the brain. And that really hasn't been done before either. But the ultimate goals of all this is to recapitulate human pathophysiology and predict human responses to drugs using clinically relevant dose exposures, not the way we do it now, where we bathe cells for many days and it doesn't scale at all to what patients see. And we had recent great example of this is we engineered a human bone marrow. To do this, we took patient-derived CD34 cells, progenitor cells from blood. We put them in a fibrin gel in one channel with bone marrow-derived stromal cells. We have endothelial cells in the other channel. We perfuse through the vascular channel, and we get a month, at least a month's worth of blood cell production. This is neutrophil maturation by FACS sorting that looks just like in vivo. This is red blood cell erythroid maturation. Gene sustain shows all different cell types. We sustain progenitor cells in these chips, and you do not get this in classic suspension culture. We could then go on to show that we reconstitute sensitivity to both radiation and drugs seen in patients. So here we're doing radiation of, we show that 2 gray totally knocks out the progenitors and of the blood cell progenitors. We preferentially inhibit the immature neutrophils and erythroid cells. But most importantly, we do this at a dose that is what's seen in patients. We then looked at drugs, and we used 5-fluorouracil, which has been shown in vitro studies with marrow cultures to inhibit their growth and induce toxicity, but it takes multiple days of being bathed in it at high dose. Patients get an infusion of 2 days with a plasma concentration of about 4 micromolar. That's precisely what we see the chips respond to in terms of toxicity response, where if you just have the same chip without flow, you don't see this. Now, the most interesting results was that AstraZeneca came to us because they had observed in clinical trials with a drug, a very unique regimen-specific toxicity where they found the same dose given over 2 hours versus 48 produced either neutropenia and anemia or just neutropenia. This is a cancer drug. Now, it's unusual for pharma to provide access to drugs in clinical trials, but this was so strange and they couldn't model it in animals. They did have the drug exposure profiles, the drug pharmacokinetic measurements. They had measured plasma levels of drug with mass spec, and that's shown in gray at the top left with the different infusions. Because we have flow, we could precisely mimic the same drug exposure profile dynamics in our chip. With that, we can mimic the same regimen-specific anemia and neutropenia, 2 hours, and only neutropenia at 48. And you could not see this in suspension culture. And we could also model recovery, which is hard to do in animals. The other thing that we did with this chip is we talked about possibility of using this for personalized medicine. We collaborated with a group at Children's Hospital who study kids with Shwachman-Diamond syndrome, which is a rare genetic disorder that leads to decreased neutrophil maturity. And what you see is when you make chips looking at low magnification, there's just less blood cell formation. When you quantify it, you see reduction of both neutrophils and erythroid cells and progenitor cells. And we can see by looking at markers of maturity that we're reducing the maturation response, particularly in the neutrophils. And this was interesting because this hadn't been seen before. And we found that we saw this in 2 patients. And then we went back to the 8 patients that they had clinical data for, analyzed them, and we found that our 2 were in a subset of patients that exhibit this syndrome. So we just discovered there's a subset of patients with a specific disease mechanism that hadn't been found before. And so this idea of being able to do potentially modeled responses for rare genetic disorders might help advance clinical trials, because it's very hard to get patients at one site for these type of trials. Lastly, early on, we had multiple drug companies come to us and say, will you make rat, dog, and human liver chips? And I was kind of against it because we were trying to get away from animal, but then they explained that every drug company has to do liver toxicity studies in rat and dog, and they also do it with human cells in a dish, and they almost always get conflicting results. And they don't know what to do. And this is one reason they kind of wing it and why there's so much failure in the clinic. And so we made human, rat, and dog chips. The chips actually have primary hepatocytes, primary liver sinusoidal endothelial cells, Kupffer cells, and stellate cells. And what you see here is one drug that we know the response from the drug companies, and that it's different in these 3 species, and we precisely mimic that on the chip. And this paper that was in Science Translation Medicine shows many drugs and many types of hepatocellular injuries mimicked using these species-specific chips. And this is something that we hope can help the companies get more comfortable with transitioning from animal models to human models. Now, we have many other chips in the pipeline. I think we have 20 right now. I'm not going to go into them all. But when I first presented this, I said that I suggest that because we have an endothelium line channel, that we might be able to develop what we call integrated human body on chips by linking fluid flow from one vascular channel to the next. So imagine an oral drug goes to the lumen of the gut chip, watch it be absorbed, metabolized by the liver chip, watch the metabolite go around, be peed out by the kidney chip. Do you have heart tox, and what does it do to bone metabolism? Or a similar drug, aerosol in the lung. And we actually have now done this. We don't use tubes to connect. There's too much dead space and it's too rigid. We use robotic samplers that can take a droplet at a time from one chip outlet to the next chip's inlet. This is great because we could change the order of the chips. We could take one drop, use it for mass spec analysis for drug levels or cytokine levels, and another drop to keep the flow going to the next chip. And it's sort of drop by drop, but we can continuously pass these from chip to chip to chip. And we showed in this paper in January this year that we had gut, liver, kidney, heart, lung, blood-brain barrier, brain, skin, bone marrow, and pancreas all linked. We could keep them alive for a month. We could actually visualize what's going on in these chips too with microscopy in the instrument. But the real goal was to see if we could model human drug pharmacokinetics and pharmacodynamics in vitro, which could help design what regimen to give in clinical trials, for example, and speed up the process. And we've now done that as well. To do that, we developed what we call a first pass model, where we have the major metabolic clearance organs. We have gut, liver, kidney, or at least the proximal tubule. We integrated an arteriovenous reservoir to mimic blood mixing, like with each pulse of the heart. So we could take samples there, like we're taking a venous blood sample, or we could inject an IV drug there and it gets distributed throughout the system, doesn't pass through one organ first. And the goal is to pull out parameters such as the maximum plasma drug concentration, Cmax, or half-life of drug, which is what clinicians need to know to design clinical regimens. We have done this with 2 different drugs successfully now. Oral nicotine, which we put through the lumen of the gut chip, watched quantified absorption with mass spec, watched it go to the liver chip, quantified metabolism of its metabolites, then had it mixed in the AV reservoir, quantified its loss in the kidney chip, and then we got levels in the reservoir over time, which are the dotted lines at the right. We did the same thing with intravenous cisplatin. Here, we don't need the gut chip because it's intravenous, but we put a bone marrow chip so we could look at pharmacodynamics, which is the effect on, biology. And again, the same computer modeling that we do, we collaborate with a company, CFDRC, that works with human PK/PD modeling. They know the masses and the volumes and the flows of every organ, so we could scale our chips to that. And here you see the dotted lines of the computer predictions from data generated on chip. What you see of the data points with error bars come from 2 past clinical studies in human patients. The top, they gave 3 different formulations and doses of nicotine in either chewing tobacco called snus, this is a Swedish study, or nicotine gum. And you can see that we precisely mimic the pharmacokinetics that have been seen in patients. Same model now with the IV drug, we precisely model the clinical pharmacodynamics seen in patients with IV infusions of cisplatin at over two different time periods. And we mimicked the pharmacodynamic effects of bone marrow suppression and also kidney injury, which is not shown here. So this now shows that this is possible to quantitatively predict human PK data in vitro. So in summary, these 2-channel organ chips we feel can faithfully recapitulate human pathophysiology. I should say that the 2-channel is critical because in the body, to do pharmacokinetic measurements or realistic drug exposure, you have to pass through an endothelium, through the matrix, into the parenchyma, and then back out again. And you really need that tissue-tissue boundary. We mimic human responses to drugs and radiation using clinically relevant exposures. We could quantitatively predict human PK, and we feel that these organ chips are now ready to be integrated into drug development pipelines. Some of you have may seen that last month or this month, perhaps 2 weeks ago, I had a review article come out, and its title is literally, "Is it time for reviewer number 3 to request human organ chip experiments instead of animal validation studies?" And in there that I review the various examples of why organ chips can produce results that if they mimic human responses, why are we having to do animal validation studies? And also integrate organoids, show the benefits of organoids, but the limitations, and that integrating organoid cells into chips, how you can get to the next level. So to end, I like to always say that, the way pharmaceutical companies work now is they'll spend millions of dollars developing a drug. They will then spend tens of millions doing a big clinical trial. They almost always fail. And then they'll do statistical number crunching to look if there's a genetic subgroup that might be more susceptible and sensitive. And if they are, they'll do a small trial and get approved for a narrow application. And that's viewed as a success. It takes years. With organs on chips and organoids from patients or IPS from patients, you can imagine searching for genetic subpopulations, let's say, 50 or 100 women with asthma who are ultra sensitive to cigarette smoke. Maybe they're an ethnic group or Hispanic or Afro-American. Very similar group. You make chips, you test drugs for them, and then you use those very patients in a small clinical trial. That could decrease the time for development, increase the likelihood of success, and greatly decrease costs. The other side of this is to use these for personalized medicine, obviously more expensive, but essentially make chips from a patient, let's say their cancer cells, their liver cells, their kidney cells, their bone marrow, find which drug is most effective for the cancer, but minimal toxicity. And there are places that are beginning to explore this, and I think this is definitely one step into the future. So to end, I can't do this without an incredible range of expertise, know-how, and brilliance in every area, and that's what the Beast Institute does. Andre Prequist is at CFDRC that does the computational modeling. I didn't describe Kit Parker's work, but he's developed a brain chip and muscle chips that we used in the multi-organ coupling. I also have to tell you, as a Harvard faculty member, I have to disclose that this technology is now being sold around the world by a company called Emulate. You could do it in your own lab. I hold equity and chair the scientific advisory board, so you basically can't trust anything I say, but I hope you believe that much of what I showed you was before we ever formed the company. And with that, I end. I invite you to our website. We truly believe that breakthrough discoveries can't change the world if they don't leave the lab. We're focused on translation. I think this is a great example, but there's a lot of other exciting stuff for those interested. So I am happy to answer questions. Thank you so much.
Thank you so much, Dr. Ingber. Brian, just a word before I introduce you. I accidentally deleted a question, and I just wanted you to be able to pull that out. Give me just one moment here. So wherever you are out there, all of you still with us, thanks. Please join me in thanking Dr. Ingber for this look at where the future of medicine is heading. And I'd like to introduce our moderator, Dr. Brian Cummings. Brian Cummings is professor in physical medicine and rehabilitation and neurological surgery at the University of California, Irvine, where he's also the vice chair for research. He's one of the founding members of the Sue and Bill Gross Stem Cell Research Center and the Institute for Memory Impairments and Neurological Disorders. His research focuses on human stem cell transplantation can affect recovery after spinal cord or traumatic brain injury. His lab is interested in factors that affect the survival of human stem cells, both in animal models and in cell culture systems. So he's located at an important juncture between the use of animal models and the alternatives that we're discussing today. So please continue to enter your questions into the Q&A box, and Dr. Cummings will field as many of these as possible. Over to you, Brian. Thank you.
Oh, thank you very much. You're welcome. So we actually have a couple of questions on the same theme, which is actually what I was going to ask you as well. So the audience is smarter than I am. They wanted to know, of course, about linking chips together that you kind of touched on, but also can you link these together to create a whole human system and then test bodily functions across the whole system?
Well, we did that in that paper. We've gone up to 10 different organs, from every major physiological system. It's incredibly hard work. It's incredibly complex. I mean, you need a team of people growing each chip, differentiating them at the right time frame, and then bringing them together at the same time, collecting samples, doing all the analysis. So, what I think is going to happen are people going to maybe link 2, 3, 4, like we've linked liver and lung to look at a drug that's a prodrug that needs to be metabolized by the liver to have activity. You might do what we did with the gut, liver, kidney to see drugs that are cleared by kidney metabolized in the liver. There are other groups that have different configurations of organ chips that are more complex and somewhat almost like explants in some ways, but Teresa Woodruff has done this beautiful model of the entire menstrual cycle and ovulation cycle, that hormones are going up and down and inducing differentiation and ovulation on a female reproductive tract in vitro. So you definitely can do that.
So another question is about how far have you gotten with the FDA? So have you or other scientists been able to talk with regulatory agencies, whether it's the FDA or UK, and get them to accept your type of testing on a chip versus using
the FDA has funded my work at quite a high level for 10 years. All the work I showed you on bone marrow, for example, and radiation is funded by FDA. FDA is incredibly enthusiastic and supportive. I've been to multiple meetings and in the European agencies where they just say, we would love to see data from pharmaceutical companies in their regulatory packages that use organ chips. But you don't go to the FDA and ask for approval of a chip. Essentially, you have to have the drug companies present data present these data to the FDA and say, we would like to use these in lieu of an animal model. And if they agree, then they basically would write, in the future, we will accept this in lieu of one particular animal model. And that we have not seen yet. There are companies, that are working on them, but we've not seen any company go directly to the FDA. But for the FDA to communicate to me that we just have to show that they're as good as an animal model, not better, just as good and human and equally robust. That's the other part, which also is not such a high hurdle because animals are not that robust, that they would be very, very supportive of that. The FDA has just announced last week a partnership with Emulate, where they're using organ chips for COVID-19, for many different, they have them on their site for 2 years already. So they're trying to get their own scientists familiar with them. So I would say the FDA is probably one of the most supportive of this of any group that we've dealt with, but they just can't start using it. People have to start coming to them with data. People, drug companies, biotech, pharma.
Drug companies aren't people, they're drug companies. But so another question is, how do you simulate these organs on a chip? Do you use software and technology or engineering first, or do you test out different designs and find out what mimics an organ the best? How do you go about this from there?
Well, there's two answers to that. One is we do it empirically first. We kind of view it as a cross section of a living organ. We try to think about starting, with the rate of breathing or the degree of breathing, the flow rates. And we've been very lucky each time it basically works. We do simulate oxygen gradients, and with all the pharmacokinetic modeling, and the organ-organ linking, we had to simulate individual organs and do measurements of drug absorption into the material, with or without cells, to every compartment, and then modeled together. And those models helped us redesign the experimental protocols, actually, flow rates and all sorts of things. So we definitely used, and that's published in the paper in Nature Bio. We had 3 back-to-back papers in Nature Biomedical Engineering in January of this year. And that is in there describing that. But we don't design an organ chip on a computer, we just build it.
So Chris Hughes, UCI, you may be familiar with him, he's asking, do your liver chips capture zonation or oxygen gradients, and how important do you think that is for drug toxicity testing?
They could, but they haven't so far. We certainly have done that in testing with oxygen gradients, and we're modeling it right now, so it can be doable. Other groups have done it and found it to be important. With organs on chips, it depends what your question is. And I often describe it as synthetic biology at the cell, tissue, and organ level. So if you can mimic clinical responses without zonation, I would say it's not important. But if you can't mimic it, then you're missing something. And one of those may very well likely be, oxygenation gradients. And I do think they're important for many things. So I think that would be a laudable goal to go at next. But as we get involved with collaborations with companies, they often have very specific, questions. Or if we're trying to get a paper published, the reviewers, want you to model a very specific thing. And, to just add more complexity for no reason is not a good way to do science. I think, it's like Einstein's approach. I mean, the simpler it is, the better. But you don't want to reduce past one molecule of problem, or else you get atoms.
All right, so Sidney Golub asks, your work is focused on small molecule therapeutic drugs. Would cancer models also work with whole cells such as CAR T or activated NK cells?
We've worked with, I showed you drugs, and we've done biologics also, we've published, we've done adenoviral vectors, we've published, we've done immune cells, that we flow through, as I showed you. Right now, Cole Roybal at UCSF is using these to look at engineered T cell delivery. I know Emulate's working with many companies looking at immunotherapeutics, but we haven't worked with CAR T ourselves.
So
No reason that would not work.
yeah, sounds like it. You'd have a system that could work. Dr. Modi asks, would CRISPR techniques in any way be useful in your research?
Yes, I mean, you know, if you were trying to model a specific gene defect, that would be a great way to do it. We've used CRISPR screens. We have an influenza model on chips. We're doing COVID-19 right now. With influenza, we use CRISPR to discover novel therapeutics that actually look like they're working against COVID-19. So, you know, it's a tool, but yes, it would be useful.
All right. And Naomi Chesler asks if you've studied the impact of different mechanics, so different membrane stiffnesses or strains, strain rate, frequency, that kind of thing.
You know, this is something that I have done in so many other experimental models. There's so much to be done. We haven't done it systematically. We certainly have done it with the gut chip. We explored different rates and degrees of distension. We were able to separate effects of flow and shear stress from effects of cyclic strain. With the kidney, I showed you that without the cyclic deformation, you don't get the full differentiation. We've shown that with the intestine chip, without the cyclic strain, you get a bacterial overgrowth of microbiome. With the lung chip, multiple, you know, physiological, pathophysiological, but we haven't gone through and systematically looked at, you know, except we did a little with the intestine chip, the degree of strain and response. In terms of the stiffness of the matrix, what people don't understand is that we play with the stiffness of the matrix in vitro often to get the cells into a phenotype that's more like in vivo. But when we give these cyclic strain, the cells remodel their own matrix. They're not stuck just, you know, to the matrix we put on the substrate. That's how they initially adhere, but they deposit their own matrix and remodel it. And I'm pretty confident that the mechanical motions are what allow them to accumulate their own matrix. We have explored getting rid of the rubber membrane with nanoscale membranes that are degradable, but we haven't published that yet. But because we could get such good clinical mimicry, that doesn't seem to be a problem right now.
So kind of combining two questions, Pamela Wynn asks, how can these chips be used to observe changes in cognitive processes as opposed to, well, either neurological ones, or someone else asks, can you model memory in one of these?
Well, you know,
are you going to put me out of business?
Well, actually, we have amazing work on that, but we don't use organ chips. We use, it's complex, but we use, it came from this, we use organisms on chips and microfluidics. We use tadpoles, and we have 1,000 chamber, looks like a peace sign, microfluidics, and they could swim. And we have electrodes, colored lights, and you could use CRISPR to engineer them. And you basically can quantify, each chip has a USB connector. We can record 1,000 individual ones swimming over time, and we can do cognitive training, memory. We've modeled autism. We've discovered new drugs that work in animals, and we're moving forward. So you have to develop different tools for different questions. And for cognition, we now do it in a high throughput way with microfluidics, but not with cells on a chip.
Right. So Dr. Modi also asked, what drug companies have approached the FDA about your organ chip model if the data is as good as the animal model?
Well, there's over 30 or 35 drug companies that are working with Emulate, you know, and we've published with, actually we published with multiple competing drug companies in the same paper multiple times, which says a lot. But, you know, we've come out with Merck, Janssen, AstraZeneca, Pfizer, GSK, you know, we published with all of them. But you don't go to the FDA and ask for, they basically have to generate data in their own labs and be convinced that it's robust enough that they're willing to say, we'd like to use these data to move this into the clinic, and to have some comparison with animal models to say it's as good or better. And they're, you know, or a company that had a drug that they never saw toxicity in animals. There is no animal model. And now they can do it on chips. That's a place where I hope maybe we'll see this go forward. The other is COVID-19, where these systems are getting integrated into BSL-3 labs. And we might get results. I mean, I've done stuff with pseudotype virus where we've found drugs that are working in animals with the native virus now, but we found them in organ chips. With the BSL-3, we're hoping to look at host response and infection. And that might be enough data with COVID-19 and the accelerated pace to get a drug through, and that could be a real help.
So could you use this system to predict the likelihood of an organ transplant rejection or graft versus host disease?
That's really interesting. So we have a lymphoid follicle on a chip. I didn't show because of time. It's published in bioarchives, where we can immunize it with fluzone vaccine, influenza vaccine, and we get the entire germinal center formation, antibody class switching, cytokine response like seen in patients, and specific antibody production. And we haven't yet explored it for graft versus host, but we think it might be usable.
All right, and a very cool question from Jeffrey Lamb. Do you consider using these organs on a chip, not just for diagnosis, but also for producing something for transplants? So like using a gut chip to correct gut microbacteria for a patient with a deficiency.
Well, we're funded by the Gates Foundation to look at probiotic therapeutics for environmental enteric dysfunction, which is malnutrition in the third world, and we have cervix and vagina chips for microbiome for vaginal dysbiosis funded by Gates that we're working on, that are literally leading to clinical trials development. But the chips are incredibly tiny, and I don't know if we would be able to grow enough in the chips for the patients. But we're certainly using it to advance therapeutics of that type.
So have you utilized organotypic endothelial cells for any of your organ-on-chip model in comparison to more generalized models for the endothelium? Have you seen improved organ identity or improved organ function?
Yeah, we have. We started with the human umbilical vein, and the kidney has kidney glomerular endothelium. The liver has liver sinusoidal endothelium. The brain has human brain microvascular endothelium. So most are organotypic. We found in some chips it doesn't matter that much for the questions we're asking. It might be very important for other questions. But in other chips, it matters a lot. Obviously, the brain microvascular endothelium, you don't get a blood-brain barrier without it. The liver, similarly. The kidney, we get slightly better effects with glomerular endothelium, but a lot of we get better transport effects, but there are other effects that we get similar. So the answer is we try to get organotypic whenever we can.
And another question from Chris Hughes again. He admits it's an impossible question, but maybe not, is how long before we do more testing on chips than we do in animals?
I wrote that review because I've literally had so many examples where reviewers have just said you can't publish without an animal mouse model. And one of them was the lung result, where we showed breathing motions affects cancer growth, and they would not let us publish unless we do an animal model. And I had to explain to the editor that it takes 3 weeks to do a tumor study in a mouse, and I can't keep the tumor growing in a dead mouse that doesn't breathe for 3 weeks before they allowed it to get accepted. I think it's going to be a while, but I think the more people voice out in the open like I did, the more we really include data that has clinical data in the paper showing you mimic it. I feel that that's hard to argue. And I think the ethical argument here needs to be heard by editors. And they ignore it, but I'm hoping more and more, the more we all do it, the better it will get.
So Lee Turner asks, I understand how data from organs on chips can be submitted to the FDA as part of a new drug application, but why can't organs on chips be regulated by the FDA as medical devices? From a regulatory perspective, are they considered tests rather than devices?
Yeah, no, they're not. A device is something that goes in your body or is used in clinical treatment. No, they view this more like a biomarker. Like, for example, KIM1 was shown to be equivalent or better than what they were using, animal studies they were using to measure kidney toxicity. And so they were able to they showed it was as good or better. They get a letter from the FDA commissioner saying, we will now accept KIM-1 as a marker for this response in lieu of animal model. And then anybody could use it thereafter. And that's how organ chip would get the stamp of approval from the FDA. It's more like more like used in the process. It's not a medical device. Medical device is a clinical device.
I'm struggling with getting the FDA to accept a prion test that's done in an amplification assay in a dish, because there really isn't an animal model to it. But as Lee Turner has a follow-up, which actually somebody just moved the question on me. But essentially Lee's follow-up was, what would happen then if they're not regulated by the FDA as a device? What would prevent a company from marketing a organ on a chip as some way to do some assay better than some other assay and not actually have sufficient data to back up that claim?
A patent. You can't market it without patents. I have scores of patents on this, and every company that's in the organ chip area has their own patents. And so, you can do it in your own lab, but you can't market anything if somebody has a patent on it, and you will be sued. And then the companies can't market it and get paid for it unless they do enough validation studies to convince the customer that it's useful. That's the answer to that.
A buyer beware. Could you use the blood-brain barrier model to test drugs that are active in the central nervous system to treat neuropsychiatric illnesses? Do we think we can move away from animal models for these types of illnesses? That was asked by Jessica Sue.
Yeah, I mean, we just got a grant with a collaborator at the Brigham and Women's Hospital, that Tracy Pierce, that she's got IPS from patients with Alzheimer's, from like 50 patients in clinical histories and so forth. And we're making blood-brain barrier chips from patients with Alzheimer's to look at the sort of neurovascular unit, which is part of the disease process, is abnormal. You can integrate neuronal cells. Clive Svensson's done beautiful work with IPS from patients with different rare brain diseases, and he's modeled them on the chips, the emulate chips. It depends what the level of molecular knowledge is. If you have some kind of reporter at the cell level, then you can use it. If it's a cognitive thing, I would use this other approach I was talking about.
All right, so our last question before we wrap up here is what you've got me thinking is that, as you've mentioned many times, it's always one company does one test with the FDA, they get approval, and it's all separated. There's no unified method here. So do you see a way for a center of some sort to work towards establishing assays and organ on a chip models that are FDA approved and kind of as a library so that people could use them without having to use animals?
It's not totally one-offs, because once one company goes and it gets a stamp of approval by the FDA, every company can use it thereafter. Anyone can use it thereafter, and it's approved. So
they have to share that data.
No. Once they share data of the organ chip that is accepted by the FDA, the manufacturer of that chip can get a letter from the FDA saying the FDA will now accept this for this disease model. So that is, it's not like each company has to do the same thing, but each animal model is one at a time being replaced. You're not replacing all mice, you're replacing a pulmonary edema model or something like that. Last week, there was a group of congressmen who put in a proposal to start a new institute at NIH focused on animal model replacements, and I gave supporting quote. So that's interesting. It wouldn't do what you're asking, but it would provide more support for it. There's a lot of support around the world. The path to how you get it approved, get FDA to accept it, I agree with you. It would be great if there's some other way to do it. They want to do it. The companies want them to do it. There's got to be some other way to do it without having to depend on this this historical path. And it is worth thinking about. The FDA is open to it. There's, different groups at the FDA, those more forward-looking, those who are skeptical and will never believe you could replace animal models. So it's not so easy to talk for the FDA, but it's possible. I think we all need to think about exploring that. I like that idea.
All right, well, I will end my moderating there and hand it back to Brianne. Thank you.
Well, thank you, Brian, and thank you, Dr. Ingber. Thanks to Jaswant Modi and the Center for Medical Humanities. And thanks to all of you who have been on the call with us today and who've contributed to this rich space of innovation between medicine, engineering, ethics, and our living planetary community. Our second lecture in this series will be in spring 2021, featuring Dr. Katrien Herman, who's a veterinarian and assistant scientist overseeing the refinement program at the Center for Alternatives to Animal Testing at Johns Hopkins Bloomberg School of Public Health. Look for a follow-up email in the next week, which will have today's video link. Also, you can find more about Don Ingber on the Wyss Institute's website. And also look out for future announcements regarding this lecture series. Thank you all again for your presence and for your energetic engagement today. I really appreciate it. See you next time.
Thank you. Bye-bye.